People are cramming. All right. Shall we get going? So the main thing we're going to talk about today is a little bit of a continuation of the previous lecture. That is, we're going to talk a little bit about how you study microbes. Finish up with what we were getting into for the topic on extremophiles, and then we're going to talk about applying many of the things that we've talked about for the first 12 lectures, including some of the recent stuff, to studies of the microbes associated with people, with various types of microbes. So I assume you all know that there's a midterm in the next second session. Good luck with that, by the way. So we were talking about these methods to study microbes, and in the context, in particular on extremophiles, we're going to move away from extremophiles, but again, continue with some of the same concepts and methods to study them. And what I want to do is go into this in detail, and I've sort of hinted at this a few times, but in detail about what we can call CSI microbiology, which is one of the methods to study microbes. So if you go to any environment on the planet, this is again the same picture I've been using for lots of things of one of the hot springs in Yellowstone National Park. But if you go to any environment, soil, water, air, skin, wherever you find microbes, there's been this interesting observation that people have made over 100 years ago, actually, where if you take part of the sample, say you take a sample of water and you split it into two equal samples, and you try and grow the things in half the sample. That is, you try and culture them in the laboratory. And then you take the other half of the sample and you just look at it in a microscope. And you count the cells that you can see in the microscope. If you do this, in general, the count that you see in the microscope is greater, usually orders of magnitude greater than the things you are able to grow in the lab. That is, if you could grow one thing from a sample, frequently you see hundreds to thousands to tens of thousands of things, of cells in the sample, and you're clearly not growing everything in the sample. So this has been called the Great Plate Count Anomaly, because the way you count cells is frequently by spreading them on one of these plates that I showed you pictures of before, and then counting the cells that grow up on that plate. And even as people developed more and more methods to grow things, so you would apply a diversity of nutritional needs and conditions to your samples, this still was true. That is, you could never grow the number of things that you could see in the lab. And this is a problem for many reasons, but one of the reasons that it's a big problem is that, as I told you, observations in the field of microbes are very uninformative as to what kind of microbe you're looking at. So if you look at the microbes in the microscope where you're doing the counting, you can see all sorts of shapes, but that doesn't necessarily tell you what they are. When you grow them in the laboratory, you can apply those methods that we talked about to studying the cultured organism, but if you can only culture a tiny fraction of the things in the environment, you're kind of stuck with understanding microbial diversity. And there are other reasons why this is challenging or important to deal with. Studying microbes, even if you could see them and identify them, what kind they are in the microscope, you might still not be able to figure out what they're doing in that environment. So for many years, people were very vexed by this. There are a few solutions that people have come up with to trying to deal with this, but the number one solution that was developed and that is used extensively today is to go to your environmental sample, rather than trying to grow anything from that sample, rather than counting anything from that sample, just grind it up, extract the DNA from the sample, and then look at the DNA in the sample as a surrogate for what organisms were in the sample. And use a variety of analyses like the ones we talked about in class about cultured organisms to study the DNA of organisms even when you haven't grown them in the lab. So you can do this for any microbial ecosystem. I'm going to walk you through how it works for what you could call a simple microbial ecosystem, and that relates to these cool organisms that I'll tell you more about in a second. So you go to this sample, you collect it from the field, you might put it in the freezer for a while, like I have a freezer full of exactly these organisms, and then you grind them up and you extract DNA or RNA or something else from the sample, but I'm going to talk to you mostly about DNA. And you take that DNA and you apply the same methods that we talked about before for cultured organisms. And I'll come back to that in a second, but I want to tell you why I picked this as the first example. So in 1977, a bunch of scientists, geologists actually, got the Alvin submarine. This is this deep sea submersible that can go down to the bottom of the ocean. And it's basically a titanium sphere that is maybe 6 or 7 feet in diameter with all sorts of equipment on it, and then a big hull on the outside of this sphere. The people go in the sphere stays at atmospheric pressure, and then the hull is the submarine, and it brings you down to the bottom of the ocean. And so they went down and they were looking for evidence for continental drift, seafloor spreading, and they thought that they were looking for volcanoes basically in the bottom of the ocean. And they had sent down temperature probes and had found sites where it was really warm. And so they went down there, this is a bunch of geologists, because no biologist in their right mind at the time would go down to the bottom of the ocean like this because there shouldn't be much of anything there. And they found this, the second highest density of biomass next to tropical rainforests. Literally piles of organisms at these sites where there were cracks in the bottom of the ocean. And what's coming up out of those cracks is warm water and various energy-rich chemicals. And after a lot of work, people figured out that this ecosystem in the bottom of the ocean is based entirely on chemosynthesis, or chemoautotrophy. There are organisms that use the energy of chemicals, things like hydrogen sulfide, to drive carbon fixation reactions, just like plants and algae use the energy of light to drive those reactions. So in the bottom of the ocean, not much sunlight there. This is 2 miles down in the bottom of the ocean where they first went. And the chemical energy is the source of everything there. Now, when people first found these giant worms, they thought, that's really weird. There must be free-living, maybe bacteria in the water that then the worms eat. They're thinking of a basis of a food chain. And people started to study these worms, and they're very weird. They have no mouth. They have no digestive system. So they're pretty strange animals when they don't have that. And actually, my My undergraduate advisor was a graduate student at a talk by someone, she was a professor now at Harvard. She was a graduate student at a talk by one of the scientists who had brought up these tube worms and was studying them. And he was saying, we can't figure out what they're doing. They have this weird giant, their whole body is like a giant organ, and it's filled with sulfur. And we think it's like a detoxifying organ. She literally got up in the middle of the talk and said, oh no, they must have these chemosynthetic bacteria that live inside of them, just like chloroplasts do in plants. And he basically said, silly girl, you don't know anything. And then in her second year as a graduate student, she had a paper on the cover of the journal Nature. And then in her fourth year as a graduate student, she had a paper on the cover of the journal Science, showing that inside these tube worms, it's a pure bacterial ecosystem. That giant organ is basically a house for things that look like this. So you look at them in the microscope, and you can see these shapes inside there that look like bacteria. Now, just like the problem with organelles, shapes that look like bacteria does not prove that they are in fact bacteria. So people spent a lot of time trying to figure out what was going on in this system. And the person, this guy Norm Pace, who sort of invented this approach to looking at DNA of microbes, this was one of the first two places that he applied this approach. So he got some samples of these tube worms. These bacteria you can't grow in the lab. So they're one of those many microbes that we are unable to culture. So that's a really big problem. They had no idea what these microbes were really doing. They knew very little about what they were. They couldn't even tell if they were archaea or bacteria. They could barely tell anything from just looking in the microscope. So what they did was they went to those samples, just like you could do with a cultured organism. They extracted DNA from those samples. So we talked about this before for cultured organisms. They ran a reaction that could make copies of the ribosomal RNA genes from those organisms. Originally, they didn't use the PCR reaction because it didn't exist yet, but they used another reaction that was much more difficult to apply, but the same principle applied. And they made lots of copies of the ribosomal RNA genes that came from the organisms in the sample. So all organisms have ribosomal RNA genes. How we could build a tree of life of organisms based upon their ribosomal RNA. And you can grind up an organism and extract out their ribosomal RNA genes, just like you could do it with organisms you can grow in the lab. You can do this with organisms from environmental samples. And so they did this. They made lots of copies of the ribosomal RNA genes. They had a method that could ignore the worm ribosomal RNA genes, so you can get copies of just the bacterial ribosomal RNA genes. But any bacteria, you don't have to know what kind of bacteria it is. And they read the sequence of those. They made an alignment, again, just like with cultured organisms, and they built an evolutionary tree. And now you can place the organisms from the sample inside those worms that you've never been able to grow on an evolutionary tree and compare them to organisms that you know something about. So this is what a tree, in fact, does look like for organisms like this. So this is from my undergraduate thesis. I was focused on another organism that actually lives on the surface that also happens to have these bacteria inside of it. So it turns out, even though these were first discovered in the bottom of the ocean, anywhere you go where there is what's called a highly reduced environment, that is not very much oxygen, basically, there are organisms there that people have described for hundreds of years that have no mouth, no digestive system, and people were vexed as to how they were able to survive. And so there's this clam that I was working on where I ran this reaction, pulled out the ribosomal RNA genes, and built a tree. And you can see in here all of these organisms at the end of their name say SYM. That means symbiont. So these are all symbionts, that is, bacteria from inside other organisms, that the ribosomal RNA genes were read without ever growing those organisms in the lab. And the most important thing here is this now tells us what the relatives of these organisms are, in order to allow us to then do studies of those organisms. We can base what we hypothesize on their relatives. So it's similar to if you go to an island and there are these giant objects on the island, it's useful to know that they're trees if you want to study them. So when we did this, we found that the closest free-living organism that had been cultured to any of these symbionts was a particular bacterium called Thiomicrospira. And it's actually, these organisms are all closely related to E. coli. They're all in the gamma subgroup of the proteobacteria. So now you can base a lot of experiments on what we know about E. coli and try and figure out what these symbionts are doing without ever being able to grow them in the lab. And this has been applied now to thousands upon thousands of samples where there's a symbiont that you can see but cannot grow in the laboratory. And that's how people have figured out an incredible amount of information about the diversity of these symbioses on the planet. So the cool thing about this method is that you can apply it to any ecosystem no matter how many microbes are there. So you can go to these other ecosystems like lakes in Antarctica, boiling hot springs, or just pond water. Do the same thing, take the sample, extract the DNA out of the sample, and get all of the DNA that corresponds to the organisms in the sample. So let's say there were 2 organisms in the sample. You take the DNA, you run this reaction that makes copies of ribosomal RNA genes. And now you have copies of the ribosomal RNA genes from those 2 different organisms. You read the sequence of those ribosomal RNA genes, you make an alignment, and you build a tree, and now you can see where organism 1 is. You don't know, you've ground up everything, so you don't know that which thing in your sample ribosomal RNA 1 comes from at this point. But you know that there are 2 organisms in your sample now. And you've classified them by placing them on the tree of life. I'm not showing, right now there are about 200 million ribosomal RNA sequences available that we can compare to. So you can get a very fine scale resolution of what your organism is in a sample by comparing to all the other ribosomal RNAs that have been characterized from other samples. If there are 4 organisms in your sample, you can do the same thing. The reaction will make copies of everything, and you can read the sequence of those 4 samples. Now, when I did my undergraduate research project, which I won't tell you how long ago it was, but it was long enough ago, it took 1.5 years to read 1 ribosomal RNA sequence from 1 symbiont's ribosomal RNA genes. We have machines in the Genome Center at Davis now that can read about 10 billion ribosomal RNA sequences in a couple of weeks. So the technology has completely changed, and that has galvanized environmental microbiology, where now people can go to any ecosystem and really survey the microbes that are present in those ecosystems by using this technology that has advanced and become cheap enough that you can do studies. So you could compare thousands of samples, and maybe processing each sample costs you $2. Whereas when I did it, 20 years ago, it cost $50,000 for one thing. It doesn't matter how many are in your sample. As long as you keep running more and more ribosomal RNA sequencing reactions, you can start to figure out and find things, even if they're rare in your sample. You might have to, so the sequences that you get out are usually reflect the abundance of organisms. So if the blue organism is present in 10 times the abundance of the other organisms, you will get 10 times as many sequences from that organism as you will from others. And that's great because now, not only do you find out who's in your environment, you can count the different organisms in the environment from their ribosomal RNA sequences. So this approach completely has revolutionized environmental microbiology. And I'm going to give you a couple of examples of what it's done. So this is one. I've shown you these trees before. I've used them in a few sittings, but I have purposefully not told you all the details on this tree. If you read the label over here, you might have figured it out. But this is an evolutionary tree of bacteria showing some 37 to 40 phyla, major lineages of bacteria. And this is the major lineages of archaea. We didn't, put you through all of these as opposed to the 6 bacteria and 2 archaea lineages. In yellow, or orange, or whatever that is, are lineages of bacteria for which at least one representative of that group has been grown in the laboratory. That is, some representative of that group has been cultured. But in blue are entire phyla for which no one has ever grown any representative of that group in the laboratory. And they are known entirely through methods like what I told you here, DNA-based methods. For microbial studies, that's sort of the equivalent if you went to an island and you couldn't see the animals or plants and knew nothing about them. This is where the vast majority of microbial diversity is, and we've been missing most of it because of our reliance upon culture-based methods to study microbes in the environment. So you can now apply these types of methods to do, oh, yeah.
I was just wondering if you think maybe the reason why some of those are not growable cultures is perhaps that these environments they live in are so extreme that they might have special requirements for sexual reproduction that isn't like what we're used to seeing. Like some very special requirements maybe that are really revolutionary to biology, but we just don't know at all how that might happen. Yeah,
that's a great question. People have tried for many years to figure out why there are some organisms that aren't culturable, and I mean, I worked on this a little bit. So the symbionts, the main theory is that the host is providing something that we haven't figured out what it is in the culture conditions. In many cases, the symbionts are like mitochondria and chloroplasts in many ways, where the host is making many things for them. For free-living organisms, you're exactly right. There's an incredible amount of diversity here, and we're applying basically methods from E. coli to try and figure out how to work with them, or from a few other lineages of bacteria. So what I'm not going to talk about is what a lot of people are doing now, which is we can also read the entire genome sequences of organisms from the environment and try and look in the data for unusual genes that don't make sense to us. We may not be able to figure out what they do, but at least we can. It's like the Donald Rumsfeld quote, we can know what we don't know in some of those samples, which at least can point you in some direction. There are about 10 or so cases now where that genomic data has led people to figure out what was missing in their growth conditions and to be able to grow the organisms. That make sense? Yeah. So you can use this general approach to now basically treat microbes in much the same way people have treated plants and animals. Still a little bit challenging, but you can at least do things like biogeography, distribution patterns of microbes across space and time. You can also do distribution of microbes across people. And this friend of mine gives me grief about this all the time. So this friend of mine wrote this review paper that used this figure that had to show semi-anatomically correct portions. And I have censored his figure because it seems somewhat inappropriate. But people have done this same type of approach on humans and surveyed them, and you can tell what are the global patterns of abundance of different types of organisms in different parts of the body. And I'll come back to this in a couple of minutes. So just keep this lovely image in your head. And I'll come back to it in a couple of minutes. But I first want to take a little break and talk in general about symbioses, which we haven't really spent time talking about sort of the general definitions, because I'm going to go through a couple of examples of different types of symbionts. So many people use the term symbioses to refer to interactions that are beneficial to both organisms. We don't use that definition. We use a much broader definition. Symbiosis is any type of intimate, long-term interaction where one member benefits from the interaction, one species, and another could have any effect on the other organism. So it's at least two different organisms, you could have more, in which at least one of them benefits. And then we can look at the different types of symbioses by simply making a chart and look at the effects on this other organism. So A, I'm saying, is the one that benefits always from this association. And then there are three different types of symbioses. Mutualism is where both organisms benefit. Commensalism is where one organism benefits and the effect on the other is basically either negligible, or frequently they use this term to refer to cases where we just don't know the benefit to the other one. It should be reserved for cases where we know that there is a negligible effect, but you'll see it in the literature all the time where it's just, so the microbes that are in and on people are frequently called commensal microbes, even though we don't actually know what many of them do. And then the third type of symbiosis is parasitism, where one of them benefits and one of them does not. So we've seen multiple mutualistic interactions already in the class. The chloroplast and mitochondria are mutualistic associations with their host. The host and the symbiont benefit there. The chemosynthetic symbiosis that I was just telling you about with these tube worms is a mutualistic association, where both the host and the microbe benefit. We'll talk a little bit about possible commensalism in a few minutes when I talk about microbes on humans. And then we've heard multiple examples already of parasitic microbes, and I'm going to give you a couple of little stories about some other ones here today where, you know, these are all the nasty microbes that cause disease, basically. So I'm now going to just walk you through 3 examples of using some of the stuff we've learned about in the previous lectures to study and learn about human-associated microbes. So the first is going to relate to these microbes that are in and on people, which we call generally the human microbiome. The biome part is sort of, you can view it as an ecosystem, like a biome in a forest. And micro is the microbial ecosystem. So these are all of the microbes that live in and on people. So if you look in a microscope, you can see microbes all over the place in and on people, lots of different places. In fact, if you count them in a microscope, you will see that there are, on most people, unless you've been bathing in antibiotics for the last few weeks, that there will be more microbial cells in and on you than there are human cells. Sometimes the estimates are 10 to 100 times more microbial cells than there are human cells on each person. Many of those microbes that live in and on people, we think have some type of mutualistic interaction with people or a commensal interaction. In other words, we think many, if not most of them, are not detrimental. But that is partly a hypothesis that is still being tested. So if you're interested in the microbes that are in and on people, you can ask the same questions that I walked you through with extremophiles. Who is out there? That is, what are the microbes that are in and on people, and what are they doing? We can use the same methods. We can observe them. We can take some of them back to the lab and try and culture them. Or we can apply these culture-independent methods, as in the DNA-based methods I just told you about. And people have applied all of these to studying the microbes in and on people for however long those methods have been around. And they have revealed all sorts of true, well-documented and or hypothesized benefits of the microbes that live in and on people. That would be mutualistic associations of some type. And this includes things like the microbes in your gut appear to be able to protect you from infection by pathogens. The microbes in and on your body are heavily involved in the development of the immune system. So if you have some disturbance in your microbial community, in particular when you're young and when your immune system is developing, there appears to be a negative effect of that in many people. Not necessarily all people, but there's a risk associated with that, and has the potential to lead to autoimmune disorders and a variety of other problems. Microbes are involved in digesting your food, producing vitamins, degradation of toxins, and I don't know if everybody would call regulating your odor a mutualistic association, but I guess that depends on what the odor is that comes off of the microbes. But there have been lots of potentially beneficial mutualistic effects shown by microbes from the culture-based approaches. In the last 5 or so years, there's been a big push to use these DNA-based methods to study the microbes in and on people. And this new push is largely because of what I was saying before, the new technology for reading DNA sequences has gone basically crazy in the last few years and gotten cheap enough that we can do this routinely to sample microbes anywhere on the planet. So why not look at the microbes in and on people? So again, there are all these locations that seem of potential interest, and this has been of such interest that now major funding agencies are developing whole programs in studying the microbiome, the human microbiome. NIH has a project that's spending about $200 million. The European Union has one. China has one. A lot of other countries have large microbiome-type projects. And from this is where this type of data is coming now. So you have some really interesting things here. So the colors here represent phyla of bacteria. The size of the pie chart represents the relative abundance of the organisms in those phyla in these particular samples. So for example, if you look here, now if you remember that figure from before, there are at least 40, if not 100 phyla of bacteria. Yet in your colon, only 2 of them are really found. The firmicutes and this other one we didn't talk about, the Bacteroidetes. So that's pretty unusual. Of all the microbes that are in and around on the planet, in the air, in the soil, in your colon, there appear to be 2 dominant lineages relative to everything else. In other places, the dominance shifts. So in your mouth, the firmicutes dominate even more. There are fewer of the Bacteroidetes. On your skin, some other lineages, the actinobacteria are the high GC gram-positives. They start to show up. And in other places, a variety of other organisms are seen. But this type of data is now finally giving us information about the microbial ecosystem that is in and on people. And will allow us to do things like compare people on different diets, compare people in different health states, compare people with different behavioral patterns, etc. The resolution is getting pretty precise. If you take a small sample, you can run this PCR reaction and make copies of even when you have just 1,000 cells or 10 cells in your sample. And so now people are saying, let's go look at a very fine scale resolution. So this woman at the NIH, Julia Segre, has been looking at a fine scale resolution of, for example, your elbow versus the top of your hand. They're different. Every place is basically a niche. And they're very different. Your elbow's, well, at least mine's pretty dry compared to, say, your mouth. And there are different microbes there, not surprisingly. Again, you can zoom in on all these different places. So much so that a colleague of mine is looking at this level of resolution. That is the left upper part of your forehead as opposed to the right upper part of your forehead. This poor graduate student of his had to submit himself to this. Actually, I think he's like the first author of the paper, so I don't think he submitted himself to anything. People are even trying to use surveys of microbes to do forensics in criminal cases. Now, there's been one, people are really trying to do this. There was one paper that got a lot of attention about this where they compared the microbes on your fingers to the microbes on a keyboard. And you can actually tell which finger is touching which key on a keyboard by looking at the microbes and comparing them to each other. This actually made it onto CSI Miami, so not the, and that's why I've been using this term CSI microbiology, because it really is in CSI. So there are some other really interesting things that people have discovered by doing these types of surveys. So if you go to the gut of different mammals, or actually I didn't really tell you, most of the studies that say colon are really of feces, because you don't necessarily want to do invasive sampling of the elephant when you're out in the field in Kenya or something to that effect. And so a lot of these studies have indirectly been trying to survey the things in the gut, but nevertheless, when you do this, people have found that across all mammals, there's an incredible similarity of the microbes that are in different mammals. So this is a tree of the microbes in humans versus mouse versus pig versus cattle, and I unfortunately don't have the overlay here, but if you overlay them, many of the exact same taxa are found in all of these organisms. And if you go back to this figure, the relative abundance of different taxa parallels across different organisms. So what seems to be going on is across mammals, there is something that helps regulate the relative abundance of different microbes in different parts of your body. It is probably parts of the innate immune system as well as the adaptive immune system that are involved in communicating and regulating the microbes in different parts of the body. And this association with some of these lineages, like firmicutes and Bacteroidetes, is hundreds of millions of years old. Again, that's based upon comparing different mammals to each other, treating the microbes as a character state, overlaying them onto a tree of the mammals, and figuring out what the ancestral pattern was for microbial relative abundance, just like you would treat any other character. There are some really interesting studies that are starting to try and understand how we get colonized after birth. So unlike the mitochondria and chloroplast, our microbes that are in and on us are not transmitted directly inside our cells, but it appears that many of them are actually transmitted during birth, during vaginal birth. People born by C-section do not get colonized in the same way as people born by normal delivery. And there appears to be a risk associated with that. Not getting colonized by your mother's microbes can be a risk factor for developing a variety of problems. And people are just beginning to study this. There are a couple people at UC Davis in the medical school that are doing some really interesting studies of colonization of microbes after birth. Yeah?
On the last chart, was there a pie chart pointing towards the vagina?
I'm sure there was.
I couldn't see on the back.
Yeah, down here. Basically, yeah. we will ignore the anatomical part of this. But basically, any orifice has been surveyed extensively for microbes. There are a lot of studies.
For science.
There's not just orifices, actually. There's, foreskin-associated microbiology, too. So there are some really interesting studies of the risk of things like yeast infections associated with what microbes are present. And this is one of the 5 areas of the human body that were picked by the NIH for their main focus. So the mouth, the stomach, the gut, the vagina, and the skin were the 5 places that are the focus for those initial studies. There are other people looking at other places like hair and etc., but those were the 5 places that were picked. All right, let's get even grosser. So if your microbial community is disturbed in some way, in particular, for example, if you have an invasive Clostridium difficile, also known as C. diff infection, there are many people for many years, although it's getting more and more popular, that have been doing fecal transplants for those people, where you find a donor who has a healthy microbial community, and they donate their microbes to a recipient. Now, usually there's some cleanup process involved in this donation where you actually isolate the microbes themselves from prior to the transplant, although I don't think that that's always the case. But this has cured multiple people of some microbial-associated problems to get microbes from a donor. Yeah?
How are they delivered? Is it surgical, or do they actually eat something?
I believe this is surgical as opposed to probiotics, which are usually consumed. And one of the problems with probiotics is they have to go through the stomach, which is very acidic and it kills most things. So I think these are being delivered to the right place. Now, this is not that surprising if you look at another kind of gross thing called coprophagia. There are lots of organisms, I'm just going to say it, that eat shit. And there have been many people for many years that have hypothesized that this is related to the microbes that are present there. So organisms that are not healthy do this more than organisms that are healthy. So that has to travel through your digestive tract, but enough might survive that it might be worth doing that. Yeah?
Are babies born with bacteria already in their intestines?
So in general, it has been thought for many years that the placenta is sterile and that the babies are completely sterile. That's not completely true anymore, so people have started to do more careful surveys, and they're definitely, even when there is not an infection in the mother, there are a small number of microbes in, like the placenta, but generally babies are almost completely sterile when they are in there. Babies can get infections, that's a big problem before they're born, but in general they're sterile. They get colonized extensively during vaginal birth. So one of the things that actually a guy at UC Davis Hospital is studying is premature infants. They get frequently born by C-section. They get put in an intensive care unit, pumped full of antibiotics, not breastfed and not touched. Not surprising, they do not get colonized with the right microbes by doing this. And premature infants are at an incredibly high risk for something called necrotizing enterocolitis. It is a horrible, horrible, invasive inflammation and microbial infection that can spread in 24 hours and kill them. And what they're doing at the hospital here is a probiotic control study to try and treat these premature infants with microbes, probiotic microbes that they eat, not being delivered directly, to see if that reduces the risk of developing this necrotizing enterocolitis. And there are, because of this NIH project on the human microbiome, there are hundreds of studies like this for a variety of ailments just starting now around the country to see what role microbes play in those issues. So I'm just going to move on to quickly tell you about the last two examples. So the first one I wanted to tell you about relates to a pathogen, that is the flu. We haven't talked a lot about viruses, but this is an example of parasitism. The flu virus is really interesting. It has its own genome. That genome is made up of 8 different chromosomes. They're actually made out of RNA, not DNA. So that's an interesting thing about viruses. Some of them have RNA genomes, not DNA genomes. These 8 different chromosomes replicate, in essence, independently of each other, just like the chromosomes in us do, but each offspring flu should get a copy of each of the chromosomes. Evolutionary analysis is used extensively to study flu, and in particular, people have now been sequencing the entire genome content of various flu strains, including each of those 8 chromosomes. And when you do this, you find something very interesting, which is that the different chromosomes within one flu do not have the same evolutionary history in many cases. And this is most likely due to recombination, or what's called reassortment, but it's a form of recombination. If you have 2 distinct flu viruses infecting one person, or one pig, or one chicken, inside one cell, the flu viruses enter. Their RNAs are floating around inside the cell. They make lots of extra copies of their genomes, and then those get packaged into new flu particles. It can mix together the RNAs from different flu strains, generating recombinants. And that is thought to be responsible for many of the major flu epidemics that have been particularly lethal over history. Because when you get this big-scale reassortment mixing different parts of the flu genome, those strains tend to not be recognized well by our immune system. They are too different from the previous year's flu strains and can lead to major epidemics. Now, I've talked about this a little bit. There are multiple theories that relate to viral origins. You can just look at this and read about it, and we can talk about it if you want. You're not expected to know this. These next couple of slides are just there for you to think about if you're interested in viruses. So I've shown a bunch of the different forms of viruses. There's a whole different set of sort of classes of viruses that have been characterized, and there's incredible diversity there. They're really completely fascinating objects. But I want to spend a couple of minutes on the last example. which many of you probably don't even remember when this happened, but this was 10 years ago now, when a particularly nasty organism called Bacillus anthracis, which is another parasite, was involved in a terrorist attack. So anthrax is a member of the firmicutes, and one of the properties of anthrax is it forms these spores that I've told you about before. Those spores make it very resistant to all sorts of things, and that's unfortunately one of the reasons that many countries have developed weapons based upon anthrax. In 2001, just after September 11th, a bunch of letters were received in 5 or 6 different places. People opened the letters and a white powder came out of those letters, and the letters had a note saying you should take antibiotics. About, I think, 5 people died from this. So they calculated that basically if a person got 2 spores of anthrax into their lungs, they could die. Germinates inside your lungs. This led to estimated billions of dollars of economic damage because people freaked out of anthrax being in the mail. And then what people tried to do, the FBI in particular, was figure out where this anthrax came from. That is, can we look at the anthrax in some way and tell something about who put the anthrax in those letters? So what they did was phylogenetic analysis. So fortunately, there was a guy named Paul Keim who was at Arizona who had been surveying anthrax strains from around the world for many years. If he went to those anthrax strains, grew them in the lab, and then tried to sequence their ribosomal RNA genes, he found something unfortunate, which was all of them are 100% identical in their ribosomal RNA genes. So ribosomal RNA genes evolve slowly. That's why we use them to study the tree of life. But if you want to compare different strains of anthrax to each other, they're useless. They are all identical in different strains of anthrax. So what he did was he then looked at a particular type of sequence in genomes called variable number tandem repeats. These were known to likely vary among closely related strains. He pulled out these variable number tandem repeats from different samples and was able to build a tree of anthrax strains based upon these VNTRs, the variable number tandem repeats. He then took the anthrax from the letters, did the same thing with the anthrax from the letters, and unfortunately when he did this, people discovered that the anthrax in the letters was most closely related to the Ames strain of anthrax. That's the strain of anthrax used in US biological defense and biological weapons labs, as well as some in Britain and some in other parts of the world. It's the main strain of anthrax used in a variety of labs. That did not make people happy. So then the question was, can we distinguish different varieties of the Ames strain of anthrax from each other that have been just passed around to different labs, so basically they've been separated from each other for a few years. The VNTRs turn out to be identical among all the Ames strains. So the VNTRs don't represent the entire genome of these organisms, so the next thing to do is to sequence the entire genome of these different anthrax strains. 6 million base pairs of DNA, and to try and see if there are any differences that have grown up when the Ames strain was sent around to different labs. So I know a lot about this because this was done at the place I worked before I came to UC Davis, a place called TIGER, the Institute for Genomic Research, and we were already sequencing the genome of the Ames strain before this happened. So then they came to us and said, can you sequence the genomes of these other flavors of the Ames strain? And that's what our institute did, and then I wasn't heavily involved in this, but I taught them how to build evolutionary trees. And they built a phylogenetic tree of these entire genome sequences, and then were able to pinpoint more precisely which variety of the Ames strain was used in these attacks. Now, it turns out that that's not the answer to everything. They could pinpoint which variety of the Ames strain, but many people had access to that particular variety of the Ames strain. So you may have heard in the last few days, there's been a controversy again. The FBI thought that they had found the person who did this. They even announced that they thought this was the person, and then Turns out it wasn't the person. And then they announced again that they thought they had the person, and it turns out it wasn't the person. And then they announced a third time, or they were charging someone, and he killed himself. And then after he did that, they announced that he was definitely the person who did it. And I mean, they have some good evidence that this guy was really involved in this. He was not a happy person. However, there was a study that just came out 2 days ago that was all over the news saying that some scientists are skeptical as to whether or not they really pinpointed that this guy was involved in it. So if you want to apply some interesting phylogenetic analyses, I'm sure the FBI is looking for help. And you can go help them figure out where this anthrax came from. Is that a Bluetooth keyboard with that?
No,
It's just a case.
yeah, it's just for the. So I had a question on this one. Like, I know, like, I went to the review session, and like when you were asking us like to do it on the floor, like I really didn't really understand that much of the topic.
Here, let me just